In my today's video, I will start a new part, that is digestion, absorption, bioenergetics and metabolism. Here are the contents of the first two items, that are digestive juices, local hormones of GIT, digestion and absorption of carbohydrates, digestion and absorption of proteins, and finally digestion and absorption of lipids. First of all, digestive juices. Definition: digestive juices are the secretions secreted from the glands of digestive tract and help in the digestion of food particles. Here is the schematic diagram of digestive system. Starts from mouth, then esophagus, stomach, gallbladder, liver, pancreas, small intestine, large intestine, and lastly rectum and anal canal. Here are the list of digestive juices. We have 5 digestive juices, that are saliva, gastric juice, pancreatic juice, bile and succus entericus, with their daily amount of secretion and their pH value. Saliva daily amount of secretion is 0.8 to 1.5 liter per day, or 800 to 1500 milliliter per day, and its pH value is 6.8 to 7. Gastric juice daily amount of secretion is 2 to 2.5 liter per day, or 2000 to 2500 milliliter per day. pH value is 1.5 to 2, and this pH is highly acidic. Pancreatic juice 0.8 to 1.2 liter per day. pH value is 8 to 8.3, and it is an alkaline pH. Bile daily amount of secretion 500 to 1000 ml per day, or 0.5 to 1 liter per day, and the pH value is 7.8. And succus entericus, the daily amount of secretion is 1 to 2 liter per day, or 1000 to 2000 milliliter per day, and the pH value is 7.5. Now I am describing the digestive juices one after another. First of all, saliva. Saliva is a viscous, opalescent or colorless fluid secreted by salivary glands. We have 3 salivary glands, namely parotid glands, submandibular glands and sublingual glands. Here are the parotid glands, submandibular glands and sublingual glands. Now the composition of saliva: out of 100%, about 99.5% is water, and the rest 0.5% is solid component. This 0.5% solid component again divided into organic components and inorganic components. Among the organic components, there are some enzymes like salivary alpha amylase or tyalin, lingual lipase and lysozyme. Second important component of saliva is mucin, then secretory immunoglobulin A, and we have some cells in saliva like yeast cells, bacteria and protozoa. Among the inorganic components, we have some important cation like sodium, potassium and calcium, and important anions, those are chloride and bicarbonate. Now the functions of saliva. First of all, mechanical function. Saliva mechanically or automatically washes down the food debris and bacteria from the oral cavity and teeth. Secondly, saliva coats the oral mucosa and acts as lubricant during eating, swallowing and speaking, and due to the lubricating function of saliva, saliva makes our eating, swallowing and speaking very smooth. Saliva protects the injury of the oral tissues and mucosa by diluting hot and eaten food. Secondly, digestive function of saliva. Salivary alpha amylase, or another name is tyalin, acts on boiled starch, and here are the examples of boiled starch, that are rice, potato and wheat, and breaks down about 30% of starch into maltose, which is an disaccharide, maltotriose and dextrin, which are the oligosaccharides. Here is the example of starch breakdown. This is the structure of starch. This is the straight chain structure of the starch, and this is the branch chain structure of starch, the enzyme salivary alpha amylase and pancreatic alpha amylase breaks down this starch into maltose, which is an disaccharide. Maltotriose contains 3 glucose molecules and alpha dextrins consisting of several glucose molecules. In newborn infants, lingual lipase initiating the breakdown of milk fats into monoacylglycerol and fatty acid. Third function of saliva, that is antimicrobial function or protective function, salivary immunoglobulin A, lysozyme, lactoferrin and myeloperoxidase enzyme control the oral microorganism growth. And saliva helps to prevent dental plaque formation by removing the adhered food particles from our teeth. Ultimately, saliva maintains our oral cleanliness. The fourth function of saliva, that is buffering function, due to the highest concentration of bicarbonate and phosphate, saliva acts as a buffer. And finally, saliva helps in our taste sensation by dissolving the foodstuffs. Now come to the second digestive juice, that is gastric juice. Gastric juice is the colorless, watery, acidic digestive fluid that is secreted by the various glands in mucous membrane of stomach. Here is the gastric gland that is present in the gastric mucosa of stomach. Now the composition, 99.5% is water and 0.5% is solid. These solid components again divided into organic components and inorganic components. Organic components like enzymes, pepsin, gastric lipase, gastric renin, lysozyme and carbonic anhydrase enzymes. And second important factor of gastric juice is intrinsic factor of Castle. Third important factor, that is mucin. And about the inorganic components, we have free hydrochloric acid, lactic acid, and among the cations, sodium, potassium, magnesium and hydrogen ion, and among the anions we have chloride, phosphate and sulfate. Here are some cells in gastric gland and their secretions. We have 3 important cells in gastric gland. Number 1, that is parietal cell, secretes gastric hydrochloric acid, an intrinsic factor of Castle. Number 2, that is peptic cell or chief cell, secretes the enzyme pepsinogen. And number 3, that is mucous neck cells, secretes mucus. Now the functions of gastric juice. We have 4 important components in gastric juice. Number 1, that is gastric hydrochloric acid. Number 2, pepsinogen. Number 3, mucin. And number 4, that is intrinsic factor of Castle. First, we will discuss about the functions of gastric hydrochloric acid. Hydrochloric acid activates the inactive pepsinogen to active pepsin. Pepsinogen acts on protein food and convert them partially digested protein food. Before the action of pepsinogen on protein food, their activation is needed, and this activation is done by gastric hydrochloric acid. Secondly, gastric hydrochloric acid maintains the optimum pH for the activity of pepsin to start protein digestion. Optimum pH is the pH at which pepsin acts maximally. And third function of gastric hydrochloric acid, that denatures the protein to facilitate protein digestion. Denaturation is a process in which primary structures of the protein remain intact, but secondary, tertiary and quaternary structures of the protein become broken down. Fourth function, hydrochloric acid releases ferric form of iron from food and enhance iron absorption. Here is the gastric lumen and here is the food iron remain in ferric form. This ferric form of iron is released by the gastric hydrochloric acid. Then this ferric form of iron reduced to ferrous form of iron with the help of ascorbic acid or vitamin C and cysteine. Then this ferrous form of iron undergo further absorption. Finally, hydrochloric acid sterilizes the upper gastrointestinal tract by bactericidal action. Bactericidal action means killing of bacteria. Now the functions of other components of gastric juice. Pepsin along with hydrochloric acid converts protein into peptone and proteose, which are the partial digested proteins. Gastric renin converts milk protein, that is casinogen, first into para-casinogen and then into calcium para-caseinate, and this calcium para-caseinate can be effectively digested by the enzyme pepsin. Gastric lipase may digest fat to some degree. And number 3 important factor of gastric juice, that is mucus, protects gastric mucosa, acts as a lubricant and acts as well as a buffer. And number 4th important factor of gastric juice, that is intrinsic factor of Castle, is essential for the absorption of vitamin B12. Now here are the process of gastric hydrochloric acid secretion. This is the gastric parietal cell. Within the gastric parietal cell, carbon dioxide is hydrated by carbonic anhydrase enzyme to carbonic acid, which is again dissociated to hydrogen and bicarbonate ion. Hydrogen ion is then secreted to gastric lumen actively by hydrogen ion ATPase pump or proton pump. Bicarbonate ion is absorbed to blood in exchange of chloride ion, which subsequently diffuse to gastric lumen and combines with hydrogen ion to form hydrochloric acid. Here are some pathological conditions related to gastric acid secretion like hyperchlorhydria, hypochlorhydria and achlorhydria. An excess of free hydrochloric acid in stomach is called hyperchlorhydria. When there is decreased free hydrochloric acid concentration in stomach, it is called hypochlorhydria, and when there is an absence of free hydrochloric acid in stomach is called achlorhydria. Now that's all for today. Okay, thank you.